So good morning to everyone. So this is the most important slide always, no? This is the people who actually do the work while I go to places to give talks. And I mean, we are in the middle of a revolution. Immunotherapy is clearly a revolution, and it owes a lot to translational research, which is the art of taking things from basic research institute to the clinic. And I happen to be working in both institutions, and actually my role is to try to build a translational bridge. We enjoy several collaborations out there, but very importantly, we have collaborations with the industry. They are producing very interesting prototypes that we test, even in first-in-human in the clinic, and also we take back questions to the lab in order to address them in the proper models. So it's getting busy, but we are undergoing a revolution because we are seeing efficacy results. And my presentation will be about immunomodulation. There will be very interesting presentations during this morning, but these ones come from a rather naive approach. And that's that in the body we have cells that can acquire the ability to kill other cells. And the pathway to get these cells going comes from quite inert cells that have to interpret very complex microenvironmental signals, recognize antigen, and come all the way to become potentially tumor killer cells, or become memory cells that can be taken back into action. And this is what we want to happen, but very often they do not respond at all. They actually get crippled and actually exhausted or anergic, or even they can die off. And they interpret signals not only coming from antigen recognition, but from a number of receptors on the surface that tell them whether to go this pathway or to die by neglect or because of exhaustion. So if you look at this, you can get all these kind of signals and divide them into positive or negative, whether they are taking the naive T cells towards the killer and memory pathway or the other way. And you can obviously use them as pharmacological targets to either enforce co-stimulation through these receptors or actually block co-inhibitions through the inhibitory receptors. And towards the end of the talk, I will be talking about CD137. That's a molecule that has been already introduced by Dr. Koss. The idea of immunostimulatory antibodies, and these things are changing our perception of tumor immunotherapy, is clearly changing. And the thing is that these are immunoglobulins that distribute in all the extracellular fluid. These immunoglobulins can block interactions that are inhibitory, or actually can enforce signaling through activatory receptors. And all the targets that are around here are already undergoing at least a phase 2 clinical trials, and some of them, such as CTLA4 and PD-1, have received approved for the treatment of a number of diseases at a metastatic stage. This one for melanoma, and PD-1 already for a number of diseases, including metastatic melanoma, lung cancer, and cancer of renal cell carcinoma, and others. And PD-L1, which B7H1 is another number for it, will probably be very soon approved for the treatment of bladder cancer, as well as probably non-small cell lung cancer. And it's very interesting because it looks like those immunostimulatory monoclonal antibodies that tamper with the brakes of the response are doing better than enforced co-stimulation. And this is kind of looking at the idea that the immune system has evolved many systems to control its function, and that those are key, and probably there are selective Darwinian forces giving an explanation for this. The first checkpoint molecule, and that's the way we call these surface receptors that control T cell activation by co-inhibition, is CTLA4, which happens to have the same ligands as the canonical co-stimulatory molecule CD28, but actually binds to them with about 10 to 100-fold more avidity, and it's able to compete this binding, thereby getting CD28 dysfunctional. And Jim Allison was the person who realized that this was actually a brake, an inhibitory receptor on immune function, And his data were confirmed by a couple of manuscripts that describe CTLA4 silence mice, and in these mice there was overt autoimmunity. So they created an antibody against CTLA4. That antibody is now called Ipilimumab, and they underwent phase 3 clinical trials, and they were successful in a couple of randomized trials in metastatic melanoma and got FDA registration. It's clearly very good to combine with cancer vaccines, as monotherapy has about a little bit under 20% overall response rate, and this comes at the cost of 30% of the patients developing some type of autoimmune reactions. When it works, this is metastatic melanoma, multiple lung metastases. It works very nice, and if you look at the dates of these CT scans, you realize that when it works, it has very durable responses. Unfortunately, in many cases, it does not work, but was clearly a milestone in setting a higher bar for the survival, the overall survival of metastatic melanoma patients. And in that realm, we thought that it was a good idea to try to incorporate these type of therapies to hepatocellular carcinoma, which could become a target for checkpoint inhibitors. And the way we did it in Spain is to try to kill two birds with one stone. And in order to do that, we chose patients with hepatocellular carcinoma that were also chronically infected by hepatitis C. And the reason behind that is that we could have a viral target in the same setting. And for that purpose, we got ongoing a 3-site clinical trial, and we were using an antibody against CTLA4, but not Ipilimumab, another antibody called tremelimumab, which has very similar biological properties. And although it was kind of underdosed in the regime that was chosen by Pfizer at that time, it clearly was able to mediate the effects. But now these antibodies in the hands of AstraZeneca, it's being developed for a number of indications, and the dose regime is much more frequent, and therefore you achieve full receptor occupancy for the whole interval between doses. So as I said, tremelimumab underwent at those doses a trial in metastatic melanoma, probably because of this underdosing and other reasons, such as using progression-free survival rather than overall survival as the principal endpoint. The trial was negative. But regardless of that, we went to this trial in hepatocellular carcinoma, in which we at least saw evidence for clinical activity, since we got 3 partial responses and many cases of stabilization of disease. This is how the first CT scan or MRR or magnetic resonance looked like. This patient eventually went to partial remission, and as you can see, the overall effects were kind of clear. And importantly enough, the viremia, hepatitis C virus, went down. In some of the patients was kind of dramatic, but typically it went to an average of about 2 logs decrease. And using a panel of antigens by the WHO that covers the whole protein sequence of the virus, as well as overlapping peptides collections, we could see by ELISpot assays that the patients under treatment were enhancing their interferon gamma ELISpot response to these viral antigens. So we were mentioning that it's very interesting to see that the immune system seems to be over-regulated. Each time we discover a mechanism that activates the immune response, we tend to discover at least 2 that control it, and there must be a reason for that. And the evolutionary reason that we can think of is placentation. In order for the species to survive, embryos with discrepant MHC, transplantation antigens, must be able to engraft in the womb of the mothers. And in that regard, it's very interesting that PD-L1 is highly expressed in the syncytiotropoblast at the surface, exactly at the interface of the mother and the fetus. And for example, IDO1, indoleamine deoxygenase, is also expressed in that region. And both these molecules are known to have immunosuppressive effects, acting locally. And importantly enough, it is curious that if female mice are carrying semi-allogenic fetuses, these are rejected if you block PD-L1 or you inhibit the enzymatic activity of IDO1. PD-L1 is kind of the star right now in immunotherapy because of its efficacy data. As you know, in the antigen stimulation, there could be these interactions, which happens to be inhibitory, or are mainly mediated by PD-1, PD-L1, or PD-L2 recognition. If there is no such inhibition, then co-stimulation and antigen receptor signaling go on more unchecked, and activation is more active. This pathway can be inhibited by antibodies against PD-1 or anti-PD-L1. And interestingly, it's kind of a more complex system in terms of receptors. The main player seems to be PD-1 combined to ligands. This one is more expressed in hematopoietic lineage, but can be also expressed in some tumors. But PD-L1 seems to be dominant in that pathway. There is another interaction described for PD-L1 with CD80 as expressed on T cells, for example, and in this case seems to be inhibitory. So it's not as clear. What the antibodies against PD-1 or PD-L1 do is to block this interaction and molecularly take away from the synapse a very important tyrosine phosphatase. These interactions that could be governed by PD-1 co-inhibition can be with tumor cells. Those are the ones we are paying most of the attention to. But it must be known that dendritic cells, other immune cells such as Treg cells, also stromal fibroblasts, under proper conditions express PD-L1, and this can be influencing the immune response as well, and probably the expression by macrophages when under the stimulation of gamma interferon and other factors could be as important. As I told you, one of the key points is that it's able to dephosphorylate substrates that go on upon antigen receptor signaling. This also happens with the chimeric antigen antibodies that we are doing. So PD-L1, by being able to carry this phosphatase SHP2 to the immune synapses, is able to drastically downregulate signaling in this directional immune synapse. So we went again to hepatocellular carcinoma with these ideas, and for that purpose we use Nivolumab, which is an antibody being developed by Bristol Myers Squibb for a number of indications, and that is able to very nicely block the interaction of PD-1 with PD-L1 and PD-L2. And this antibody has, as monotherapy, a broad spectrum of clinical activity that goes in overall responses up to 33% in melanoma and to a lesser extent in other indications. There is some correlation with the amount of mutations that are computed in the exomes of the tumor cells and the ability to respond, although this is not a strict correlation, and there are clearly exceptions along this route. For example, non-Hodgkin's lymphoma refractory, you have something like 70% of response with very little mutation, although in that case, the locus that encodes PD-L1 and PD-L2 is clearly amplified and overexpressed in the red stem cells that are the malignant cells in that disease. So we had several evidence that PD-1, PD-L1 were in the microenvironment, and also mouse data telling us that PD-1 blockade could be useful in models of hepatocellular carcinoma. So we wrote and started this as an investigator-initiated trial that eventually was sponsored by Bristol Myers. We reported this in ASCO last year, and it was a very careful dose escalation with 3 etiologies for hepatocellular carcinoma, including an infected hepatitis C and hepatitis B. We were extremely afraid of precipitating severe hepatitis, hyperacute hepatitis, fulminant hepatitis, but it didn't happen in any of the patients, and that's the reason we started with very low doses and did a very carefully ascending phase all the way to overdose to 10 milligrams per kilogram, since the standard dose across the program and the one we are using in extensions is this one. This trial has been amended several times. Now it's going up to 250 patients with several exploratory cohorts and has regulatory value because potentially can drive for approval for second-line hepatocellular carcinoma. This is the kind of patients that we reported for the escalation phase. They were typical patients with preserved liver function. We only treated the child-pug A's and the B's, only the B7. And it was clearly, there was a Asian heritage was dominating hepatitis B due to the epidemiology of this disease in Hong Kong, which was one of the centers that participated early on. So all the patients had received previous treatment, either local approaches or most of them had received first-line sorafenib, and those that were included without sorafenib were a minority, were because of being intolerant or because of having declined this treatment. And the overall response rate by RECIST 1.2 criteria was a little bit shy of 20%, which is kind of remarkable in this population, observing some complete responses that, as you will see, are durable. Interestingly enough, responses went all the way irrespective of the etiology of the hepatocellular carcinoma. So there were responders in the infected and the virally infected cohorts. And I want to draw your attention to a fact which is common to many diseases treated by PD-1 or PD-L1 blockade, and that's that we get a lot of patients with durable stabilization of disease that we believe are going to be driving factors for the survival curves. When partial responses occur, sometimes they are very dramatic, as this case in our clinic. And we were commenting the stem cell, well, with some detail how to remove inhibitors, but our favorite game is to enforce co-stimulation. And for that regard, we are very keen of the molecule CD137. This is a TNF receptor a family member that is expressed typically on activated lymphocytes. It has been described elsewhere, but it's clearly expressed on activated T cells and NK cells that have met antigen either through the antigen receptors in the case of T cells or CD16 engagement by ADCC on NK cells. And there is only one described ligand that is expressed on activated antigen-presenting cells. That's 4-1BB ligand or CD137 ligand, and that conveys co-stimulatory signals to prime T cells very nicely. And all came from a kind of very simple experiment that we performed already many years ago in 1997, and that's to use agonist antibodies against CD137 in mice bearing transplanted tumors, some of them with the reputation of being poorly immunogenic. And in this case, we were able to see complete rejections of some of these tumors, leaving immune memory and protection for rechallenge. And we clearly saw that a CD8-mediated immune response was the main factor behind those rejections. But over the years, we have found roles for multiple cells that mediate these effects. And for example, we know that dendritic cells were required probably for cross-presentation of antigens, as you will see. And also we found something very funny, is that tumor endothelium in the vessels of the tumor is actually expressing ectopically CD137, and agonist ligation on the vessels actually creates their homing receptors for integrin ligands and chemokines that facilitate the entrance of T cells into the tumor tissue. It's debatable whether Tregs are being influenced by CD137. They clearly express this molecule in the tumor microenvironment, but we don't have a clear picture yet. And it's very interesting because CD4 cells also express the molecule. You can co-stimulate CD4 cells in vitro. In vivo, it looks like that they are repressed after transient activation by the antibodies, but not by the natural ligand. We don't understand this very well, but it's interesting because the same antibodies that induce the rejection of mouse tumors actually ameliorate autoimmune conditions in mouse models of autoimmunity. We figure out that the main signaling pathway underneath 4-1BB was TRAF2 mediated, and TRAF2 is a ubiquitin ligase enzyme that creates polyubiquitin lysine 63 polymers that are docking sites for other signaling pathways driving to NF-kappa B and to MEK activation. Now we have another story on this, and that's that these polyubiquitin chains that happen to get induced in several substrates are actually regulated by deubiquitinases such as 820, SEAL, and probably others that are present in the complexes and are able to inhibit this. In the field of immunotherapy with immunomodulation, probably the most exciting news is that you can go to combinations achieving synergistic effects, and that those are therapeutic effects in which the final effect is better or stronger than the sum of the individual effects of each agent. In this regard, we try to go through strategies that would get something going on. You start the engine, you get something prime, and you can do that by several things. There is a spontaneous responses against tumor in many cases that you can use, the baseline response, and then kind of supply the co-stimulatory signals and remove the inhibitory signals with immunostimulatory monoclonal antibodies. Several of these strategies are being tested in the clinic in combinatorial approaches. We favor a lot the idea of turning some of the tumors into a vaccine by manipulating locally a tumor in such a way that with proper adjuvants, it will present the whole array of antigens that it's there. And combination immunotherapy looks very complex. The arrows represent the combinations, whether there's evidence for synergy at the preclinical, that if it's already in clinical trials or has been approved, as is the case of anti-PD-1 and CTLA-4 that has been approved for dramatic effects against melanoma at the expense of undesirable toxicity in about half of the patient. There is another one that has not been published yet, although was communicated in SITC, and that's PD-1 blockade inhibition together with IDO inhibitors, indoleamine deoxygenase inhibitors, that is probably looking very promising. To test this combination, sometimes you have to go to spontaneous models of cancers in gene-modified animals, and we got one which a double transgene bidirectional, controlling the expression of the c-MYC oncogene and ovoalbumin as a surrogate tumor model under a liver-specific promoter that controls the tetracycline operon. And in this setting, the animals that are not drinking tetracycline develop a multifocal hepatocellular carcinoma that happens to express by immunohistochemistry ovoalbumin as a surrogate tumor antigen. And in this setting, we test our favorite triplet combination, which is anti-CD137, anti-PD-1 or anti-PD-L1 blockade, and anti-OX40. OX40 deals with regulatory T cells nicely and is a powerful co-stimulator of CD4 responses, and is the joint function of CD137 promoting CTLs that probably gives rise to a very potent synergy that is much better in terms of survival of the mice than the doublets or the singlets of these treatments. And it's very interesting because in this model, regardless of expressing ovoalbumin in all the malignant cells, if you infuse these mice with transgenic T-cell receptor cells that recognize ovoalbumin, both CD4 and CD8, OT1 and OT2 cells, plus generose interleukin-2, you have very little effect or none. The combo 3 was again having this kind of very interesting prolongation of survival, but it's extremely interesting that this triple combination of immunostimulatory monoclonal antibodies plus adoptive T-cell therapy now has a very exciting synergy in terms of controlling tumor growth that goes up to the lifespan of the animals. So this is kind of really remarkable and telling us that combinations are very interesting. And one of the points behind combinations and synergy is that there are pharmacodynamic interactions. And this is very clearly shown because now if you look at the tumor-infiltrating lymphocytes and what are these cells doing under therapy, you realize that by providing one of the immunostimulatory monoclonal antibodies, you change the level of target expression for the other antibodies. For example, if you provide anti-CD137, PD-1 goes overexpressed as well as PD-L1, so you need combinations to make this work. In this case, CD8 cells were the primary dominating factor in the response because if you deplete them, you lose the effect, and you can see the CD8 cells in the tumor with a smoking gun because they have the killing machinery, and there is evidence that they have degranulated since lysosomal proteins are on the plasma membrane. So there is a synergy with adoptive T-cell therapy that can be exploited, and actually in some tumors, such as B16 ova and OT1 cells, can be extremely dramatic if provided early on. All the animals get basically cured, although if you delay this, it's not as clear. But this dramatic synergy, because monotherapies do nothing, and the combination is extremely powerful, in this case, it's combination with anti-CD137 single treatment, permits to go and do mechanistic experiments, such as the requirements of CD137 by using CD137 knockout mice that we made, or having that cross to the T cells. And we find that both in the endogenous animal, in the endogenous T cells of the animal, as well as in the adoptively transferred T cells, CD137 expression was a requirement. And this was correlated with the presence of a more intense infiltrate in the tumors, not only of the transfer CD8 T cells, but more importantly of the endogenous CD8 cells that also get access into the tumors. These cells are more active in terms of showing activation markers, showing degranulation and intracellular interferon gamma. And if we perform multiphoton in vivo microscopy of chamber tumors in animals under treatment, it's possible to see that there is this response in the tumors with a slower delay in the time of observation. We could use OT1 cells that are fluorescent, as well as the tumor cells with a red a histone labeled in red to be able to monitor the chromatin and the nucleus and check the combination. And what clearly changed our mind about how these things are working is that we realized that under the influence of anti-CD137 antibodies, the OT1 adoptively transferred T cells behaved in a different fashion. They moved less quickly, they stayed on the targets until they do their job. So now if this video works, it will show you that under the influence of CD137, the trajectories are kind of shorter and they stay on a single target. And if you follow it longer, you can see that they stay long enough until they disrupt the chromatin of the target, meaning that they are more active interactions, giving rise to the death of tumor cells. So probably there is a kind of very important marriage in immunotherapy of CD137 and adoptive T-cell therapy. This has been used in different ways. You can select out the best T-cells in a culture by selecting those activated good enough to express CD137 on the surface. If you co-stimulate the cells in vivo for adoptive transfer, they perform better after having received signals through CD137 that they seem to remember. You might know all the stories from the labs of Carl June and also from Stan Riddle and Brenners in Memorial Sloan Kettering that show that chimeric antigen receptors recognizing surface antigens on T cells with a single chain antibody are very successful in the treatment of B-cell-derived malignancies. The most successful ones have the signaling domains of CD137 in their cytoplasmic tail, and this is critical. We know this story of stimulation of some of the tumor endothelial cells in the capillaries, giving rise to more infiltration of the T cells, and there seem to be in vivo microscopy portals of entry that are actually kind of hotspots for T-cell entry into the tumors. An important question is whether the antigen is being directly presented by tumors or by antigen-presenting cells. For the activation of CD8 cells, it's becoming clear that there are subpopulations that are specialized in this job. In the human, they express CLEC9A and CD141, and these are kind of professional cross-priming dendritic cells because they can deviate uptaken antigens to the MHC class 1 antigen presentation pathway. And these cells are characterized by a chemokine receptor, XCR1, that drives them to interact with CTLs, with prime T cells, and with NK cells. And these cells for development are critically dependent in a single transcription factor, which is BAP3. So the BAP3 knockout mice have a few of these cells, and those that they remain there
are kind of dysfunctional. And they are known to produce large amounts of IL-12, particularly if stimulated through TLR3 or TLR7, and also by the cytoplasmic receptors of RNA, such as MDA5. And what was very interesting to find is that the treatment with CD137 or PD-1 antibodies, or the combination of the two that is highly synergistic against this colon cancer, which is MC38, is totally gone in these animals that have a defective, which are knockout for BAP3 and are defective for these cross-priming dendritic cells. And interestingly enough, since they are necessary, probably you can stimulate them to get synergy. And if you increase their numbers by providing their favorite growth factor, which is soluble FLT3 ligand, and you inject poly-IC as a surrogate of double-stranded RNA into the tumors, you get synergy, and that synergy is totally lost in the knockout animals. We need to model this in order to be able to tell things about combinations, and for that regard, seems to be a useful idea to get animals repopulated with human immune system and bearing tumors and be able to perform experiments in this setting. So we were able to do that thanks to a xenografted tumor from a gastric carcinoma patient who is now with no evidence of disease following surgery and local radiotherapy, and who is still donating some PBMCs to us. And by getting RAC2 IL-2 gamma receptor double knockout mice and grafting the tumor and also the PBMCs from the patient, we could see that PBMCs mediate some kind of tumor control, but this is very much enhanced if the animals are co-treated with either with monotherapy with anti-CD137, the same antibody that we are using in the clinical trials, and Nivolumab. And this effect is very clear. And if you take out the tumors at the time of sacrifice and perform multiplex immunostaining to be able to visualize the target of the treatment, PD-1 and CD137, you can see that the T cells are there. The CD8 T cells attack the tumor from the periphery in that setting, and you can actually tell them apart. And the animals have higher concentrations of human interferon gamma circulating as a result of providing these immunostimulatory monoclonal antibodies. So our gratitude, as always, goes to patients and their families, very brave people allowing us to develop new immunotherapies as we learn. And you know, learning is a complicated process in which you are doomed to make some mistakes. Centrally, there are central facilities in our institution that have helped a lot, and these are the people who are financing our work. So thank you very much, and I will be glad to take questions, because without these people, we won't be able to build a bridge. Thank you very much.